Glycolysis is the metabolic pathway that converts glucose into pyruvate and, in most organisms, occur in the liquid part of the cells. The free energy released in this process is used to form the high-energy molecules known as adenosine triphosphate, or ATP, and the reduced nicotinamide adenine dinucleotide. So, glycolysis. Etymologically speaking, glycolysis came from the two Greek words glyco, meaning sugar or sweet, and lysis, meaning breakdown. So it is basically the breakdown of sugars. Glycolysis starts with a 6-carbon molecule known as glucose, and it is where in the cytoplasm where glycolysis starts. It starts with 2 ATP molecules being added to the glucose to form fructose diphosphate. The fructose diphosphate will then be added with 2 NAD to produce NADH and 4 ADPs to create 4 ATPs. And then the end product will be the 2 pyruvic acids which will proceed to the citric acid cycle. The entire process of the glycolysis produces at least 4 ATP molecules.
So now we will talk about how does acetyl-CoA form. So it will start in the process which we call pyruvate oxidation or transition step in cellular respiration. So we all know that pyruvate is the final product of glycolysis. So it have the 3 states. So if the oxygen is present, it will go the pyruvate oxidation. And now the pyruvate oxidation have 3 stages. So the first is the pyruvate molecule will go to the cytosol and transport into mitochondria. So the pyruvate is composed of 3 carbons. Now in pyruvate oxidations, 1 large pyruvate molecule will musulod na siya sa mitochondria. So mudri na mag-start ang first stage na to. So which we call the decarbolization, which kanang ang isa ka carbon kay ma-remove siya. Then, musulod na siya sa secondary stage, which is ang decarbolization kay ma-oxidize siya. Means sa oxidize is loses electrons. Mo ni siya ang NADP+. But ang kaning NADP+, kay mag-reduce, then mag-gain siya electrons na pod, which is the NADH. Then, we all know the acid molecule kay mo-undergo sila sodium hydrogen, which is maon ni siya ang H+. So, karon ang kaning decarbolization na molecule na na-oxidize siya, kay magstay ni siya dire o makarry siya sa coenzyme A. So what is coenzyme A? Coenzyme A is a vital molecule that's involved in numerous biochemical reactions and protein, carbohydrate, and lipid metabolism. Its main function is to carry acyl group such as acetyl stereoesters. It's an essential part of our body's functions. So ang coenzyme A mo ni siya ang magbind sa all molecules, then dire na siya mag-form ang acetyl-CoA. Malnish. Pag ma-carry na to siya sa coenzyme A, katong mga decarbolization na na-oxidize, kaya din na siya ma-form ang acetyl-CoA. So, muna siya ang large pyruvate molecule na ma-undergo sa first stage, which is the decarbolization. So, tapos, ma-oxidize siya dire sa secondary stage. Then, i-carry siya sa 3 stage, which is the coenzyme A. Then, pag mabind na ang all molecules, dire na mo-form ang acetyl-CoA.
The citric acid cycle, also known as the Krebs cycle, is an essential component of cellular respiration in living organisms, taking place within the mitochondria. This cycle involves a series of chemical reactions that systematically break down acetyl coenzyme A, or acetyl-CoA, derived from the breakdown of carbohydrates, fats, and proteins. The cycle generates high energy molecules, including NADH and FADH2, which carry electrons to the electron transport chain, contributing to the production of adenosine triphosphate, or ATP. Not only does the citric acid cycle serves as a crucial energy producing mechanism, but it also plays a pivotal role in recycling molecules for further cellular respiration.
Electron transport and chemiosmosis, presented by Jessa Altaya Paipa. We've examined the first two steps of cellular respiration. These are glycolysis and the citric acid cycle. As it turns out, neither process generates much of an energy payoff, but the NADH and FADH2 that were generated in the citric acid cycle move on to oxidative phosphorylation, which generates by far the most ATP out of these pathways. These pathways utilizes an electron transport chain, which is a series of mitochondrial membrane proteins that sit in the inner membrane of mitochondria. And we refer to these as protein complexes 1 to 4. These proteins bear a variety of prosthetic groups, which are non-protein components that give the protein its functionality, including flavin mononucleotides and cytochromes. There is also one component in an electron transport chain that is not a protein, and that's ubiquinone, a small hydrophobic molecule that is mobile within the membrane, and it is also known as coenzyme Q or CoQ. Once NADH feeds electrons into the first component of complex 1, these proteins facilitate a series of redox reactions, shuttling the electrons downhill from one component to another, with each structure down the chain having a higher affinity for electrons than the last. This process does not generate ATP directly, but a byproduct of this series of electron transfers is the generation of a proton gradient across the membrane. Protons accumulate outside of the inner mitochondrial membrane, which then go to power another protein complex called ATP synthase. As you might guess from the name, this is the component that synthesizes the ATP. Because the proton concentration becomes greater in the intermembrane space than inside the mitochondrial matrix, the protons will spontaneously move with the proton gradient to re-enter the mitochondrial matrix, and the only route available is through ATP synthase. This process is called chemiosmosis, and because the gradient is able to do work through chemiosmosis, we also call this proton motive force, and it is these protons that power ATP synthase in phosphorylating ADP to generate ATP.
Aerobic organisms. So what are aerobic organisms and how do they differ from anaerobic organisms? Well, from the word itself, aerobic, which has the Greek roots aero meaning air and bios meaning life, simply means that aerobic organisms are life forms that thrive with air. While aerobic organisms are microscopic prokaryotes or eukaryotes that use oxygen as a catalyst, such as bacteria, protists, or yeast to produce energy. Certain aerobic microbes use oxygen to survive. While some creatures or life forms can live without oxygen, others still opt to use it. Numerous earthly creatures fall into the aerobic category, meaning that they utilize oxygen for the synthesis of the cellular energy, such as mammals, frogs, fish, birds, mosses, plants, and insects. These are only few types of aerobic organisms.
Everyone, my name is Jobis D. Casus, and I will explain to you what are the obligate aerobes, the facultative anaerobes, and the microaerophils. These three are all types of bacteria that are aerobic. Aero means with air, and they are called aerobic bacteria because they consume or use oxygen to make ATP. Some specific examples of bacteria that fall into these categories are the Mycobacterium tuberculosis, the Pseudomonas aeruginosa, the Bacillus subtilis, and the Escherichia coli. Anywhere on Earth where there is enough oxygen such as most of the terrestrial regions and the upper oceans, is home to aerobic life. There are no aerobic creatures in environments devoid of oxygen.
And now we will discuss the characteristics outlined for anaerobic organisms. Number 1, anaerobic organisms do not use oxygen. Anaerobic organisms function in the absence of oxygen, so it highlights their adaptability to environments with limited or no oxygen available. So this stands with a contrast to aerobic organisms which rely on oxygen for their metabolic processes. Number 2, H2O and potassium nitrite are the byproduct. While aerobic organisms only have H2O as the byproduct, anaerobic organisms have H2O and potassium nitrite as the byproduct. The metabolic pathway of anaerobic organisms result in the production of water or H2O and potassium nitrite. So these byproducts provide insights into the chemical transformation occurring during anaerobic respiration.
Anaerobic organisms with electron transport chain. Anaerobic organisms are organisms that can thrive and generate energy without the need for oxygen. Unlike aerobic organisms, which rely on oxygen for their electron transport chain or ETC during cellular respiration, anaerobic organisms use alternative electron acceptors. In anaerobic respiration, these organisms utilize substances other than oxygen as the final electron acceptor in the electron transport chain. Common alternative electron acceptors include nitrate, sulfate, and carbon dioxide. Despite the absence of oxygen, anaerobic organisms still undergo a process similar to aerobic respiration, involving glycolysis, the citric acid cycle, and an electron transport chain. The specific electron transport chain in anaerobic organisms varies based on the electron acceptors used. For example, in sulfate-reducing bacteria, the electron transport chain involves transferring electrons to sulfate ions. In the absence of oxygen, anaerobic organisms have evolved diverse mechanisms to adapt and generate energy for alternative pathways. In simpler terms, anaerobic organisms are those that don't need oxygen to survive. Unlike us, they use different substances instead of oxygen to generate energy. In their version of the energy-making process, called the electron transport chain, they use alternative things like nitrate or sulfate instead of oxygen. This helps them adapt and thrive without relying on oxygen. Anaerobic organisms, electron acceptor is nitrate or sulfate. In anaerobic metabolism, when oxygen is unavailable, organisms resort to electron acceptors like nitrate or sulfate during respiration. Nitrate reduction, for instance, involves the stepwise reduction of nitrate to nitrite and further to ammonia or nitrogen gas. Similarly, sulfate reduction results in the production of sulfide. These processes enable anaerobic organisms to extract energy from organic compounds, sustaining their life cycles in environments where oxygen is limited or absent.
Anaerobic organisms that occur in prokaryotes and requires no special organelles include bacteria and archaea that thrive in environments with low or no oxygen levels. These organisms have evolved to carry out anaerobic metabolism without relying on specialized cellular structures like mitochondria. Instead, they utilize various metabolic pathways to generate energy in the absence of oxygen. Examples of anaerobic prokaryotes include Clostridium species, which are known for their ability to produce toxins and can ferment organic compounds to produce energy. Methanogenic archaea are another example. They produce methane as a byproduct of their metabolism in anaerobic environments such as swamps, marshes, and the digestive system of animals. These anaerobic organisms demonstrate remarkable adaptability and metabolic diversity in environments where oxygen is limited or absent.
Fermenting organism. Fermenting organism is a microbe or organism responsible for the process of fermentation. In simple terms, it is the agent that causes the fermentation of organic substances, such as like sugar through fermentation. Through fermentation, compounds like sugar are converted into various products like alcohol and lactic acid. First, do not use oxygen. Fermentation is an anaerobic process, meaning it occurs in the absence of oxygen. Fermenting organisms do not use oxygen as their primary electron acceptor during cellular respiration. Instead, they rely on the other molecules or ions to accept electrons generated during the breakdown of organic compounds. Second, lactate or lactic fermentation or ethyl alcohol or alcoholic fermentation is the byproduct. Final acceptors of electrons are pyruvate reduced to lactate and acetaldehyde. Lactate fermentation and alcoholic fermentation are two common pathways employed by fermenting organisms to regenerate NAD+ and sustain glycolysis in the absence of oxygen. In lactate fermentation, pyruvate serves as the final electron acceptor and is reduced to lactate, a process that regenerates NAD+ from NADH. These mechanisms allows organisms like certain bacteria and muscle cells to maintain glycolytic energy production under the anaerobic conditions. Alternatively, in alcoholic fermentation, the final fermentation acceptor is acetaldehyde, which is reduced to ethyl alcohol or ethanol. This pathway is utilized by yeast and some bacteria, including those involved in the production of alcoholic beverages. But lactate and ethanol production are crucial for facilitating anaerobic energy metabolism, enabling cells to sustain glycolysis and produce ATP in the absence of oxygen.
No electron transport chain. Unlike cellular respiration, fermentation does not involve an electron transport chain. ATP is generated through substrate-level phosphorylation. Occurs in prokaryotes and eukaryotes. Fermentation is a metabolic pathway that occurs both eukaryotic and prokaryotic cells. Simple and faster alternative to cellular respiration. Fermentation is a simpler and faster alternative to cellular respiration. It allows cells to produce ATP without the need for oxygen and the complex processes of the electron transport chain.
Requires no special organelles, unlike cellular respiration, which occurs in the mitochondria of eukaryotic cells. Fermentation does not require special organelles. It can occur in the cytoplasm. Glycolysis and waste product formation are two sets of reactions that occur. Fermentation involves glycolysis, the breakdown of glucose, as well as the subsequent reaction that lead to the formation of waste products such as lactate or ethyl alcohol.
So for the similarities, both involve carbon dioxide, CO2, is a waste product of cellular respiration in organisms. It is also a byproduct of combustion processes in various human activities like burning fossil fuels. In cellular respiration, electrons are transferred from glucose to NADH. This process occurs during the breakdown of glucose to produce energy, involving the transfer of electrons through a series of reactions in the electron transport chain.
So hello guys, so for today's video, I'm gonna talk about the similarities of these three organisms, which is the aerobic, anaerobic, and fermentation. So before that, like magstorya sa tower, istoryahan sa nato kung unsa jud ba ning tulo. So like the conditions, process, ATP production, ana. So dit merit. Aerobic. Conditions, no? So requires the presence of oxygen. Process involves the complete breakdown of glucose in the presence of oxygen. Location takes place in the mitochondria of eukaryotic cells. And ATP production yields a large amount of ATP. So up to 38 molecules of ATP per molecule of glucose. So sa aerobic. So the second one is the anaerobic. So conditions occurs in the absence of oxygen. Process partial breakdown of glucose without the use of oxygen. Location it happens in the cytoplasm of the cells. ATP production ATP production generates less ATP compared to aerobic respiration. 2 molecules of ATP per molecule of glucose. So sa anaerobic respiration. Fermentation, conditions takes place in the absence of oxygen. So same sa anaerobic. Process involves the partial breakdown of glucose without using an electron transport chain. Location occurs in the cytoplasm. And ATP production produces a small amount of ATP, usually 2 molecules of ATP per molecule of glucose. So same sila dun sa anaerobic. So in summary, all these processes are mechanisms for cells to generate ATP. So it means that ATP is a crucial molecule that stores and transfer energy within the cell. And despite the variations in the efficiency of ATP generations, these 3 organisms has a fundamental goal, or the fundamental goal across these organisms, and that is to produce energy for the cellular function. So that's all. Bye.